name is Michaela Whipple, and today I'll be presenting my landmark presentation over the paper titled Selective In Vitro Growth of T Lymphocytes from Normal Human Bone Marrows. This was published in 1976 by Morgan, Russetti, and Gallo in the Journal of Science. So this paper made a really critical contribution to our understanding of how T cells, which are one of the most important immune cells, how they can be grown outside and studied outside of the body, not just inside. So it really helped pave the way for later breakthroughs in immunotherapy, cancer treatment, and the discovery of key immune signaling molecules. Okay, to begin with, we need to look at the scientific environment of the early 1970s. So to begin with, T cells are very important for our adaptive immune system because they're responsible for recognizing and responding to specific antigens. So within this T cell group, there's two main types. There's the helper T cells, which are going to assist our B cells and macrophages, and then there's also the cytotoxic T cells, which are going to directly kill infected or cancerous cells. So these two types of cells end up actually working together, and they coordinate and execute our immune responses. Second off, the bone marrow is known as the body's blood cell factory. It produces B cells, red cells, and platelets, and then other components of the blood that we all know. However, back in the 1970s, there was very limited understanding of the T cell development in the bone marrow, so most of it was just focused on the thymus or the peripheral blood. Third, there were very technical limitations in the 1970s, so it was difficult to isolate and grow T cells in the lab, especially from bone marrow. Researchers didn't have reliable methods for selective T cell growth, and on top of that, key growth factors like interleukin 2 or IL-2 hadn't even been discovered yet, so we really didn't know that these help support T cell survival and growth. Lastly, because of these limitations, there was a clear scientific need, so researchers couldn't study T cells in detail, which would really help progress immunology. To better understand how T cells develop, respond to pathogens, and function in disease, they needed a way to reliably grow T cells in vitro from a clean and sterile bone marrow source. And so that's exactly what this problem in this landmark study aimed to solve. Okay, to fully understand and appreciate this study, we need to go over some key concepts that led to it. First off, the role of T lymphocytes. So these are very important in our adaptive immune system, like I already mentioned. And I also mentioned that there's the helper and the cytotoxic T cells. So the helper is going to assist other immune cells, and then also the cytotoxic are going to kill infected or cancerous cells directly, and this is known as cell-mediated immunity. Next, there were some major challenges in T cell research before 1976, so although scientists knew that T cells were important, they had a very difficult time growing them from bone marrow. This is because there were limited methods for selectively growing T cells in vitro, which meant that researchers couldn't study them in isolation and under controlled environments. Next, researchers began exploring the idea of soluble growth factors, which we now call cytokines, that actually help T cells grow. At the time, these factors hadn't been clearly identified yet, but the study was a part of that early research in discovering them, and also the interleukin 2, which turned out to be one of the most important T cell growth factors. Lastly, a tool called PHA, or phytohemagglutinin, was used to stimulate T cell activation. PHA is a plant-derived mitogen that can non-specifically activate T cells, and this encourages them to divide and grow. So this study used PHA as a key part of its method to stimulate T cell growth during culture. Altogether, these concepts really helped shape the direction of the study and helped the researchers to design a really good strategy to overcome the technical barriers that they were facing in T cell research. Okay, so for the hypothesis, the researchers asked just a very specific question, and this was, can T lymphocytes be selectively cultured from human bone marrow? This was a very significant question because if they could end up controlling T cell growth in vitro, it would open up several research possibilities in immunology, therefore oncology and infectious disease. So their hypothesis was specific conditions, including mitogen stimulation and growth-promoting factors, will allow selective expansion of T cells from bone marrow. Now we'll look at the methodology that the researchers used in this study. They started by collecting bone marrow samples from healthy human donors. And bone marrow, again, is a source of all blood cells, including immune cells. So it was a very smart place for the researchers to start to try and grow T cells from. And to activate the T cells present in these samples, they used a mitogen called PHA, which again stands for phytohemagglutinin. And PHA is derived from plants. It's a protein that stimulates T cell division and activation. It's commonly used in lab settings when researchers want to encourage T cells to proliferate. And next, they added something called a conditioned medium, and this is a culture fluid that has been used to grow other cells and therefore contains various secreted factors like cytokines or growth signals. And although they didn't know at the time, this medium also probably included interleukin 2 or IL-2, which we now know is essential for T cell growth. And finally, they monitored cell growth over time and used surface markers to identify whether the growing cells were actually T lymphocytes. So these steps allowed them to confirm that their method was both effective and selective in expanding T cell growth from the bone marrow. The results of the study were clear and significant. So first off, the researchers did achieve successful and selective expansion of T cells from the human bone marrow samples. This was a major breakthrough because it showed that T cells could actually be grown reliably under the right conditions. Importantly, non-T cells like B cells or myeloid cells did not grow in the same cultures. So this confirmed that the method was selective for specifically T lymphocytes, which was exactly what the researchers were aiming for. And the study provided proof of concept that functional T cell cultures could be established directly from the bone marrow. So it demonstrated that the bone marrow contained T cells that could be activated and grown. And this helped lay the groundwork for many future discoveries in immunology, including how T cells develop and function in the immune system. Because of these significant results of the study, it had a long-lasting impact on the field of immunology and medicine. First off, it really laid the foundation of the discovery for interleukin 2, which again we now know is a really important cytokine for T cell growth and survival. Interleukin 2 was shortly identified after this study, and its discovery was directly linked to methods developed in this study for culturing T cells. Second, the ability to selectively grow T cells really opened the door to T cell cloning, which allowed researchers to generate large populations of similar T cells for experimental purposes. And this helped us understand antigen specificity, immune memory, and T cell signaling. Third, these breakthroughs were essential for the development of adaptive immunotherapy. This is where patient T cells are modified or expanded outside of the body and then can be reintroduced to fight diseases like cancer. This method also became important in HIV therapy and research, where understanding and being able to manipulate T cells were key in managing viral infection. Overall, this study marked a major advancement in hematology and cancer immunology. It gave scientists the tools to study and eventually take T cells in ways that can continue to save lives today. So, as I look back on this study through a Christian perspective, I am reminded of Psalm 139:14, which says, "I am fearfully and wonderfully made." The immune system is very complex and very organized, and the fact that our bodies can distinguish between what belongs to us and what doesn't, and then also train immune cells to protect us, just reflects a higher and intelligent design from a creator. So I believe that this complexity, therefore, is not random, but rather it is purposeful. So each immune system interaction, each signal, and each cell type that plays a role in contributing to our larger immune system was designed for our protection and healing. So as Christians, we are called to care for what God has created, and that includes using science and medicine to care for others and ourselves. So research like this really helps equip us with the knowledge to treat disease and develop therapies and make important moral decisions about how we can apply this scientific discovery. So ultimately, science becomes a way to not only understand God's creation deeply, but to also fulfill our calling to love and serve others through healing. So, to conclude, this paper by Morgan, Rossetti, and Gallo really marked the first successful in vitro expansion of T cells from human bone marrow. And this was a groundbreaking achievement in 1976 because it overcame a lot of technical barriers. And it also used a new method of using PHA, and that allowed scientists to study T cells in a more controlled and reproducible way. So, this methodology became a model for future T cell experiments and really helped establish new lab techniques in immunology to make new discoveries. So, it paved the way for the discovery of interleukin 2, again, a cytokine that drives T cell growth and survival. And this discovery led to even more important clinical applications like cancer immunotherapy or HIV treatment. So, this paper is a great example of how basic scientific research can have a ripple effect across decades of research and science. And this shapes what we know about diseases and how we treat them, and just about the body's design as a whole. And this is my references page. Thank you for taking the time to listen to this.